So in the print project, in the different case study sites, 7 to 37 compounds were applied. A total of 180 different pesticides were applied in the 10 case study sites, and 23% of the applications exceeded the recommended application dose. And per field, we had a maximum of 19 to 20 pesticides that were applied. So that means farmers and as well the environment do not only via the application, not only come in contact with one single pesticide, but with a mixture of pesticides during the application process in the growing season. I will now show you what we found in the different matrices. We start with soils. So 99% of all, we compared here, I forgot to say this, conventional and organic farming systems. And AR means Argentina, because what I show you here is a number of residues and the range of them, the median in the different farming systems and in Europe and compared to Argentina. What we found out that 99% of all conventional farm soils contain pesticide residues and 96% of them contain mixtures. What is tested now by EFSA in the pre-approval assessment, only single pesticides. So this does not at all reflect the reality. Even 95% of the organic farms still had at least one pesticide residue, 79% contained mixtures. The number of residues in Argentina was lower than in Europe because in Argentina they concentrate on less pesticides, but these applied in a higher dose. In the crops, that's where we could see that mainly the crops, most of the crops were below the maximum residue levels, but most of the crops as well contained mixtures, and finding until a maximum of about 20 pesticides per plant. In the air, we as well detected all mixtures. We here installed passive samplers that was staying 2 months in the fields during the growing season to see which pesticides are in the air, which residues are in the air in organic and conventional farming systems. And here we didn't find any difference. Dirk and Daniel and Freya will explain later on why, because these pesticides are transported over long distances. And so, but here's where we find always mixtures with up to more than 25 different residues. And now we come to the indoor dust. The indoor dust, we detected in the highest, we detected 124 different pesticide residues in indoor dust of farmhouses. The lowest amount we found is 25. And this means that residents who are living there are permanently exposed to pesticide residues through indoor dust. The risk is not assessed until now and not included into any EFSA or, as far as I know, EPA risk assessment regulation. In water bodies as well, mixtures were always present. And Nelson will tell you in the last presentation today, what does it mean for aquatic life. The concentrations here to be seen is, you see here a logarithmic scale. So in soil, crop, air, water, and sediment, they were more or less the same concentrations, but in indoor dust, we found a maximum of more than 200 milligrams per kilogram pesticide residues with farmers' houses. Just because of this discussion now on the different concentrations in the different matrices, you can see here the different pesticides we tested. The approved ones here are only shown. And in the left block, you see the conventional, the ones under conventional, and the right, in the middle one under organic farming. The right one is in water and in sediment. And the less dark then the point is, the higher the concentrations are. So what you can see here, you see here frequencies and the concentrations. So what you can see, all pesticides which were approved are detected with high concentrations in indoor dust. And this as well is especially true for the ones which are absorbed to the soil and then transported with wind. And you can see here this, especially for AMPA and glyphosate, in indoor dust, they highly accumulate the risk for inhalation of humans or ingestion of dust is not known. Now we come to a discussion or to do a very clarification of terms. What is hazard? What is risks? A hazard is something that has the potential to harm you. And the risk is the likelihood of a hazard causing harm. That means I first of all will now show you the hazardous characteristics based on EFSA conclusions and the PBDB database. So what are the characteristics of the residues we have detected in the different environmental matrices? I give you a small selection from. So you can see here on the left side the approved pesticides that are still approved on the market, the candidates for substitution, which are supposed to be phased out, and then we have some non-approved old pesticides, mainly DDTs, DTEs, and so on. First of all, we compared the residues detected in soils. Are they described by EFSA to be hazardous for earthworm reproductivity? And the darker the color is, the more hazardous they are. So what we find out is, even from the approved ones, nearly 80% are moderately toxic for earthworms. And for the candidates for substitutions, it's about 90%. And from the not approved ones, there's a lot of no data because these data were not tested when these products were on the market. What about fish? 5% of the products we detected in water bodies are highly hazardous for fish, and 67 or 80% moderately hazardous for fish. And here as well for conventional and organic farming, taking into consideration the residues detected in outdoor air. So we could see that for conventional farming, 18% are high to moderately affecting honeybees, but 65% are not. Known data are missing. And for organic farming, then there were no high toxic substances, but as well moderate toxic substances. So that means the residues we detect in the environment are mainly moderately hazardous for the terrestrial and aquatic ecosystem, following EFSA conclusions. We did the same exercise to see what are the hazardous characteristics for the residues detected in indoor dust of conventional and organic farmers. Left side again, approved candidates for substitution and non-approved ones. So what we could see here that 3 plus 33% of the products which are approved on the market are yes or possibly cancerogenics. And this amount is higher for candidates for substitution and non-approved ones. 85% or 86% of the mutagenous products are mainly phased out from the market. The neurotoxic as well. For endocrine disruptors, we saw that 23% of the approved ones are yes or possibly endocrine disruptors, and it's for both systems, conventional farming houses or organic farming houses. And there's a lot, nearly half of it, we don't have data for this. And for reproduction development effects, we could see that 60% of the residues we detected in indoor dust for the approved substances are yes or possibly having a negative effect on reproduction development. And for the candidates for substitution, it's even higher, and for the non-approved ones as well. So that means we are surrounded by residues which are potentially harmful for our health. But since the EFSA or the EPA do not take into consideration the intake of indoor dust, and there are no benchmarks for actual daily intakes, we do not know which is the risk. I just would like to show you now one result which I get today from my colleague from Cork, who are working as well in the Sprint project, and just giving you an example how we can link now hazard to risk. What they do in Cork, they test the effects, and here we only show the results for glyphosate on the actual daily intake, ADI, on the behavior of mice. And what you could see here is that even at the actual acceptable daily intake rate of 0.5 milligram per kilogram per day, the mice, you could see here on the left side, the mouse in the corner of the kind of box, the mouse who is exposed 6 weeks to the acceptable daily intake by glyphosate, and these mice nearly do not go away from the corners of the box, whereas mouse, the control mouse, who is not exposed to this glyphosate, nearly is curious and is walking around in the center of the box. So there, my colleagues from Cork see strong changes in behavior, and they're related to anxiety-like behavior. They did similar experiments to see what is the effect of social interaction, and then they find out for glyphosate that the acceptable daily intake, there is no effect, but if you get for higher doses, you can find an effect. There are studies ongoing now on the effects of, on the risk effects of in rat experiments and cell experiments. Some will be presented today. I will just present you here as well an idea of prioritization of pesticides related to ecosystem health, because the farm to fork strategy of Europe is now searching a possibility how to phase out 50% of pesticides and which ones should be phased out then. My colleagues made here a prioritization based on occurrence of the different residues in the ecosystem and multiplied them by the hazard. And so you can see here this beautiful log transformed graph. But what is important, you can see here above the pesticides which are following this approach, the ones which should be phased out because of hazard and occurrence in the environment. They did the same exercise for human health, and here you can as well see the products which should be phased out then to decrease the human health effects. My take-home message for you. Pesticide residue mixtures are omnipresent in ecosystem and humans, highly accumulated in indoor dust. Most of the residues are hazardous for ecosystem and humans. The real risk for ecosystem and humans being exposed to mixtures of high numbers of hazardous pesticide residues is not taken into consideration in regulations. EFSA regulations do not include atmospheric transport as a related risk. My colleagues will talk about this in some time. What we need is pesticide regulations that assess real risk, including sensitive indicators, a transition to a more sustainable and 2030-ready agriculture system based on agricultural concepts, and therefore all stakeholders need to be on board to achieve a transition towards reduced pesticide use. Thank you for your attention, and sorry for the chaos at the beginning.
Thank you very much, Violette, for your presentation. Okay, very, very nice and interesting. Are there questions here in the sale? Do you have questions here? Okay, if not, then we go online. Is there any question there online? Can you please check, Celia?
Yes, I'm checking. No, there's nothing in particular.
Okay, there is one from German Lenner. Please, German.
Hi, can you hear me?
Yeah.
Yes.
Hi, a very nice presentation. I am a researcher from Argentina. I am currently studying the simulation of pesticides and mixtures and something like that. And I wonder if in Europe you have some kind of regulation or mandatory laws, because it seems that here in Argentina is kind of a freedom in terms of application and spreading out of pesticides, and maybe it's really related with the results that you show. So my question is, if in Europe you have more strong regulation or not?
They have clear regulations. I was recommended pesticide dose that should be applied, but what you could see, even following farmers' interviews, 23% of the pesticides are overused. 20%, yeah. So we have regulations, but our regulations here in Europe, they try to regulate, but there's a lot missing to really face the real world and to simulate the real world conditions. I think we have stronger regulations in Latin America, but there's still a lot.
Okay, thank you very much.
Thank you.
Thank you, German. So we go to the next presentation. We have Abdallah Alloui from the University of Uber, and also he go deep more into the Spring project. Please, Abdallah, the floor is yours.
Okay, yeah, thank you very much, Esperanza. The title of my presentation is How is exposure to pesticides affecting ecosystem, plant, animal, and human health? An assessment based on Springfield Company across Europe and Argentina. My name is Abdallah Alloui. I am working at the University of Bern in Switzerland. And for this work, I was supported by different experts and participants of the Sprint Consortium. The objective of this study is to assess the association between exposure to pesticides and environmental, plant, animal, and human health, defined here as EPA health. And the specific objectives are to characterize EPA health in conventional and organic farming systems, to list the most frequently found active substances across environmental and biological matrices, and to assess their potential hazard. Sampling, as mentioned by Violette, was done during the growing season of 2021 in 11 case study sites, based on a monitoring plan in which we provided standardized methods for sampling, storing, and laboratory analysis. We analyzed a total of 209 substances of a total of 4,375 samples, and from the three compartments: human, animals, and environment. For human, we considered three groups: farmers, neighbors, and consumers. The different matrices assessed for this purpose as are listed in the second column here. First results regarding the 10 most frequently found substances. When considering all compartments and matrices of a total of 4,375 samples, the 10 most frequently found substances are DDE, PP with 49%, followed by glyphosate and folpet, and you can see the following substances on the table on the left side. Similarly, we classified substances within each compartment. You can see also that DDE-PP is found in all compartments with high percentage from 49 to 65%, followed by glyphosate and folpet PHA. This is only an example of the 30 most frequently found substances within these 10 substances. In the next slides, I'm going to show some important results regarding the frequency of each matrix of each compartment. And I am providing some examples. Here, DDEPP occurrence, which is a metabolite, was found in all matrices with high frequency in the environment in conventional as well as in organic farming system. The most important results consist of its occurrence in the blood of the 3 human groups with a very high frequency. Relatively high frequency was recorded for animal 2, especially in feed and feces. Also, for glyphosate, second most frequently found in all matrices, we note its occurrence in all matrices with very high frequencies and its occurrence in human blood of farmers and neighbors, while in the consumers there was no analysis. Piperonyl butoxide was found in water and in indoor dust with high frequency in conventional as well as in organic farming system, and also in all matrices of animals and human, including plasma of all human groups. Tebuconazole was found in almost all matrices with the highest frequency in indoor dust, as you can see, and with relatively low frequency in blood of the 3 human groups, neighbors, farmers, and consumers. Fipronil sulfone was also found in indoor dust and human blood with high frequency and with moderate frequency in the animal feces. Similarly, we found hexachlorobenzene in all matrices of the environment, but also in human feces and blood with relatively high frequency. Here we establish the relationship between frequency and potential hazard. For convenience, we selected 3 classes from low to high to match the classes of the evidence level for human and ecosystem class of hazard given in the plant protection database. The red boxes on the top right side represents the most potentially hazardous class since it corresponds to a high level of evidence in terms of hazard and a high frequency of occurrence. What we can deduce from this, in addition to the hazard of each substance, is that if we move from conventional to organic, hazard remains same, but the frequency of its occurrence decreases. This is the case, for example, for the blue and the green boxes in the middle and left side. We have drawn touch tables for the 10 remaining most frequently found substances that we are going to provide in the paper in preparation. For the ecosystem, we assess the risk for single pesticides based on the risk quotient defined as the ratio between the measured environmental concentration of the given matrix, MIC, that you can see on the top left side, and the predicted no effect concentration, PNEC. The risk quotient provides a measure for risk across different ecological matrices. Here are soil, plant, water, and sediment. For the 4 matrices, we provided the risk of each substance and the related frequencies, how many times the related risk occurs. Orange, for example, in the table represent the high frequency of occurrence, more than 50%. In the case of azoxystrobin, for example, a risk in soil was classified as medium with 15.6% for the conventional and as a negligible risk for organic farming with 6.3%. And we can see that in red column on the left side present the 13 found substances in all matrices with different risk coefficient and related percentage. Here I am giving the list of the substances that are present in at least three matrices, soil, plant, water, or sediment. There are 9, and we have here 7 substances found only in 3 matrices, soil, plant, and water, and the 3 last one are found only in one matrix, water or in sediment. In summary, in this study, we highlighted the 10 most frequently found substances in all compartments and matrices and their potential hazard in EPA, ecosystem, plant, animal, and human, and their risk defined for ecosystem. DDE-PP is the most frequently found in both conventional and organic, followed by glyphosate. Indoor dust, as mentioned before, contains almost all the substances analyzed at high frequency on both conventional and organic farms. The substances were more frequently found in soil of conventional farms than in organic farms. Some substances have been found in human blood at high frequencies, such as the DDE-PP. Glyphosate was found only on conventional farms. The other substances, as peperonyl butoxide, tebuconazole, fipronil sulfone, hexachlorobenzene, were found on both conventional and organic farms. In animal, all the substances were found either in urine, feces, and in feed. Thank you very much for your attention. If you have any question, please feel free.
Thank you very much, Abdallah. Very nice presentation. Are there questions here? Okay. No, there are not. Online, do we have questions? Can you check, Celia, please?
No, there is no questions.
Okay, thank you very much, Celia. So, we have one here. Okay, yes, please, can you give just a moment? Do you hear the question?
No.
Okay, so can you please go? Yes, the microphone is not working well. So here we have Patricia Aki from IFAD. So please, can you approach, please?
Sorry, quick question. I just wanted to ask what they're doing in Latin America with regards to local farmers who are selling their farm or products on the streets to help them with.
I cannot hear you very well. Sorry.
Oh.
Hello?
Do you hear now?
Yes.
Okay. Well, the question of Patricia is related to Latin America.
I just wanted to ask what you're doing in Latin America with regards to helping farmers sort of cleaning their vegetables and their produce before they sell it out to the consumers.
Esperanza, can you help me, please? Repeat the question.
Yes, thank you very much, Patricia. So the question is, if in Latin America, do you know what are they doing for helping farmers in relation to this problem?
No, I am sorry. I have no clue about Latin America, but because I'm working in Europe.
Mm-hmm.
And the project is linked to Argentina. I don't know if some participant can help replying this question. Maybe Violette, I don't know.
Okay, we can go further with the with the presentations, and then we keep your question, Patricia. So we have your email, so we can also be in contact with you and give the good answer. So then we go to the next presentation is from Daniel. Thank you very much, Abdallah. So then we go to the next presentation, that is Daniel Figueiredo from the University of Utrecht.
So I'm going to go now from one of the matrices, which is indoor dust, to human exposure of residents living close to conventional and organic farming. Now, you might ask yourself, why indoor dust? Well, one of the things is from different scoping reviews and literature reviews, it's the less understood route for pesticide exposure. It is easy to be also exposed to pesticides in dust via ingestion or contact with surfaces, inhalation of, for example, their suspended particles. You also can think of that humans spend most of their time in indoor environments where dust accumulates. And most importantly, when we look at children, infants, and toddlers, and crawling always with the ends in the ground, taking the ends to the mouth, that can lead to higher exposure. Now, if you see this image that Violette presented, you can see that several pesticides were found in indoor dust comparing to other matrices. These are not the only reasons. I can give you a few more. For example, Zeng saw that there was significant exposure risks relating to, for example, dermal contact with contaminated beddings and furniture, while Koskola saw that for most pesticides were actually accumulated in ultrafine and coerced particles. What does this mean? Is this particle size can actually penetrate the lungs. Moreover, the dust residence time, this means that the time that dust takes to completely be out of your residence, can go up to months to 7 decades. So it can be quite persistent. And finally, in a recent study, soil ingestion was estimated to contribute 46 to 78% on hand-to-mouth ingestion of children in Bangladesh. So these are just several reasons why we should focus more on this indoor dust. Now, the methodology in Sprint, in short, we looked at 168 indoor dust samples. Majority were from where farmers were living, 128 samples. These homes were located close to conventional and organic farms across Europe and also Argentina. We analyzed, as was mentioned, 198 pesticides and one synergist, and we used these measured pesticide concentrations as input to estimate exposure via dust ingestion. Now, let's look at the results. What you see here is an overall picture of conventional on the left side and organic on the right side, split by fungicide, herbicide, and insecticides. At a glimpse, you might say, well, there's actually not that much difference between conventional and organic. There are some differences, but these are varying per pesticide. What I can mark here is, if you look at the y-axis, that's about 10 micrograms per gram of a pesticide per dust. This is higher than non-farmer worker ohms that was previously studied in the Netherlands. This is also higher than non-farmer worker ohms from a previous study done in California. We saw that comparing to a study in 2012, they also detected similar pesticides, even some that were banned, such as chlorpyrifos banned in 2020. And similar concentrations were measured for certain pesticides in indoor dust from urban ohms in East Taiwan, indicating that there is this persistence in dust that we saw in Sprint, also seen in other studies. Now, if we move from concentrations to exposure, what does that mean? Well, in simple terms, exposure from dust ingestion is the amount of pesticide in dust times the amount of dust you ingest divided by body weight of an individual. Let's take, for example, chlorpyrifos in Sprint project, and this is the range from 0.83 to 7,000 nanograms of chlorpyrifos per gram. Let's take the average weight of an infant, 5 months female baby, 6.9 kilograms, and a realistic estimate of exposure that 5% of the infant population takes their hand to mouth and leads to ingestion of 0.14 grams of dust per day. If we calculate, this means that exposure from dust ingestion was about 0.14 micrograms per kilogram. You might ask, what does this mean then? What is the threshold here? Well, previously set toxicological value of 1 microgram per kilogram, known as the acceptable daily intake, was set for chlorpyrifos in 2014, meaning that for this pesticide and for dust ingestion in this scenario would contribute for 14% of the daily intake. You might say, well, that's about 1/5. Yes, but I'm only talking about 1 exposure route and 1 pesticide. So just imagine all the pesticides we analyzed in this study, and not just dust ingestion, all the other routes, such as dietary intake or inhalation. So let's move on. How does this actually compare with other routes? Well, first, before we go into that, there is one question that is in everyone's mind. How does it compare between conventional and organic farming? This is what we're seeing here. Just looking at the trend, let's not look at the y-axis, concentration, or the x-axis, different pesticides. Looking at the trend, conventional versus organic, overall, residents living close to conventional farming were higher exposed. Now we add the y-axis. Well, that is about 0.003 nanograms per kilogram body weight, which it's quite low, but it's not always the case. Let's take another example, and here I emphasize on glyphosate, which some of you might have heard of, that conventional in this case was on average 10 times more exposed than living close to an organic farm. And if we look at exposure, this is actually 1 nanogram per kilogram body weight. So orders of magnitude higher. Now, I'll show you an example of the different routes that contribute to total exposure. So we're going to compare dust ingestion with other routes. This is an example for Denmark, but if you see our deliverables, this actually translates to the same for the other countries. What you'll see is that 1 bar is 1 participant. And what I'm showing you here is that we look at total exposure from 0 to 100, 100 meaning total exposure from all different routes. So if you plot this, what we see for glyphosate, in this case, we have our farmers, we also have different populations such as neighboring consumers. And what we see is that in some cases, exposure was actually predominantly from the dust matrix, while in other cases was from food intake. We saw this for not just glyphosate, but several pesticides. Actually, exposure was predominantly the environment and not diet. And as I mentioned, the results are similar for different countries. Let's look at another example, which is the Netherlands. In this case, I'm putting dust ingestion apart and looking at another route, inhalation of resuspended particles. In this case, if we look at the inhalation of resuspended particles, we see that this route is predominant over the other routes. This stresses the importance of environmental exposure over dietary exposure. Now, this brings me to my take-home messages, which are the following. We analyzed dust samples and we saw that there was up to 120 pesticides in ohms close to both conventional and organic farming. Certain pesticides, such as glyphosate and AMPA, were considerably higher when compared to the other measured pesticides. And pesticides in indoor dust can be a dominant route for exposure when comparing to other routes. This, of course, depends on the pesticide properties, spraying amount, if there was an indoor use. So based on this, my recommendations are that we need an urgent revision of current European non-dietary risk assessment, which is quite limited, more focused on dust ingestion and inhalation routes. Given that this matrix is quite important, we should look especially at children's exposure and study more the pesticide levels, the mixtures, and the driving factors. And with a range of so many pesticides, we have to ask the question, what does this mean for effect of these pesticide mixtures present in indoor dust? We need more toxicological studies. And finally, just as a recommendation, frequent cleaning and leaving shoes outside at the doorstep can reduce your indoor exposure. Previous studies also shown this. Thank you very much.
Okay, thank you very much, Daniel. So are there questions here? No? Okay, so online, do we have questions?
Online, we don't necessarily have a question, but we do have a comment from Biomata. saying that in a wide study some years ago, most residues were found in crawling babies.
I would like to put attention on this issue, that we have in Europe a precautionary principle. That means the governments are obliged to protect citizens against unwanted risks. So it is very in consideration with this precautionary principle, it is very important to take into consideration this fact that Daniel said, this environmental exposure of people is not taken into consideration when we look at acceptable daily intake. So we urgently need action here to get this included into regulations.
Hello.
Yes, biomatter. I guess you want
to say. Thank you. This is Ahmad, and I don't have a option for raising hand. I don't know why. That's what I just came online. If it's okay, just
yes,
or
reactions.
Yes, okay.
Yeah, okay. Other than that I mentioned some years ago, from the spraying in home that they use it, unfortunately a lot, ladies. Yeah, they found a lot, most residues in crawling babies. Also, in my research 40 years ago, in my master research residue analysis by GC, wide research, we found that anyway, children are generally more susceptible to pesticides because of the lower maybe cytochrome P450, the detoxification enzyme system, and other things. Thank you.
Okay, thanks a lot.
Thank you very much, Amanda. So we go to, thank you very much, Daniel. So we go to the next presentation, please. We have Hans Moll from the University of Wageningen.
Okay, thank you. See if I can share my screen.
Yes.
Now,
yeah,
okay.
Now it's ready. The floor is yours, Hans.
Yes, thank you. Okay. So you see the screen in presentation mode? Not yet, I guess. Yeah. Okay, thank you for the introduction. I'm going to talk about pesticides in human urine and feces. And the first question, of course, why do we want to do that? So we have heard in the previous presentations that pesticides are around in the environment, different compartments, especially dust also. So that's an external presence, and we have a potential exposure from there. Of course, this is only an issue for humans when we have an uptake of these pesticides, either oral, dermal, or through inhalation, so that it results in an internal exposure. And by measuring the pesticides in urine and feces, we get an insight in the actual internal exposure. So, and this process is called human biomonitoring. So it's a method for assessing human exposure to chemicals by measuring the chemicals, in this case, pesticides or their metabolites, their derivatives, in human specimens. And these human specimens can be excreta, like urine or feces, but it could also be blood or hair. But in this presentation, I focus on urine and feces. Well, from external to internal exposure, as already mentioned, there are different routes. Of course, pesticides are used as plant protection products, and they may leave residues in food, so we will have dietary intake. They may end up in the air, attached to particles, so we can have inhalation if you live close to these fields. The pesticide can be present in dust, as just discussed. And it's also important to notice that some of the pesticides are also present in other types of products, for example, in biocides that are used in households or for vector control. And some of the pesticides double as veterinary medicines, so in these flea collars or in livestock. So there's different sources and there's different routes of intake. And with human biomonitoring, we get an idea about the aggregated internal exposure through all these routes and all these sources. Now, the currently used pesticides in the EU, but basically worldwide nowadays, are non-persistent and not bioaccumulative. So what goes in at some point will go out, and then mostly through urine and feces. So if we measure those matrices, we get an idea about the actual exposure to pesticides and also to which pesticides we are simultaneously exposed. This picture you have seen, samples were collected in 10 case study sites in Europe, 10 different countries linked to certain crops, and 1 case study site in Argentina. So there were 4 subpopulations sampled: conventional farmers, organic farmers, neighbors, so people living close to the farm fields, and control or consumers that live away from these agricultural fields. Almost 800 participants included, and we had a very high response rate. Over 90% returned their set of morning urine and feces samples. Now, a little bit more on the matrices. First, urine as a matrix for human biomonitoring. For most pesticides, pesticides are excreted relatively rapidly from the body upon uptake, which means what we see in the urine reflects exposure from the past 6 to 24 hours. It also reflects the actually adsorbed fraction. So those are pesticides that have entered the bloodstream and may have reached the organs. Sample collection is easy. Urine is the most common HBM matrix. The analysis is a bit tricky. We typically have a relatively poor coverage of the number of pesticides that we can measure in urine. And the most important reason is that most pesticides are not excreted as such in the urine, but they undergo metabolism. And I show you here for tebuconazole what happens to this compound in the body. It is metabolized into multiple different derivatives. And in the human, in this case, there are 2 that are dominant and can be found in the urine. But you can only identify and quantify them when you have the analytical reference standard. And for many of those metabolites, that is not the case. So, and this limits our possibility to cover a wide range of pesticide metabolites in urine. The existing data, partially for that reason, is mostly focused on pyrethroids and organophosphorus pesticides and some others. In Sprint, we try to extend it to some 44 pesticides and their biomarkers, plus a dedicated method for glyphosate and AMPA. So we needed several methods, which I will not discuss in detail here. It's also good to mention that not all the compounds we measure in urine are specific for a certain pesticide. In some cases, a compound that you measure in the urine may have originated from different parent compounds. And I give an example here for pyrethroids. This 3-PBA compound we measure in the urine can come from a whole range of these different pyrethroids. And the same thing is for these other biomarkers. They may come from different pyrethroids. It's only DBCA that is one-to-one linked to delta methrin in this case. Let's have a look at what we found in urine. And here I show the detection frequencies. There are 2 colors of bars, blue for what we found in the EU, and orange what we found in Argentina. And of course, Argentina was only 1 case study site, so certainly not representative for the whole country. And on the y-axis, we see the percentage of samples in which a pesticide was detected. And here I show you the top 20 detected urinary biomarkers in the EU, 10 EU countries. Number 1 was TCPI, and TCPI, all the red ones, by the way, here are metabolites, and the black ones are the parent compounds. TCPI is a common biomarker for both chlorpyrifos and chlorpyrifos methyl. It was basically found in all the samples, both in Europe and in Argentina. 3-PBA and some of the other non-specific biomarkers for pyrethroids were found in high numbers of sample. Some of the phenoxy acid herbicides, 2,4-D, quite a number in the EU and in almost all samples in Argentina. And we had glyphosate and AMPA as well in many samples. We see that if you compare, there are some limitations, of course, but if you compare what we see in Europe and Argentina, that for some detection frequencies in Argentina are much higher than in Europe. But that's not always the case. There are also pesticides for which it's the other way around. For example, tebuconazole, much more frequently found in EU samples than in this one site in Argentina. But it clearly shows that you can see differences between different regions. That were the individual pesticides. We also had a look at the mixtures. And in many samples, we found multiple pesticides or multiple biomarkers for pesticides in the urine. And here we see the median number of pesticides per sample, per case study site, represented here as country, and also different color bars for the different subpopulations. So the Netherlands, Argentina, Portugal were amongst the countries where we had the highest number of pesticides in the sample. Slovenia and Denmark were lower. We also see some differences. For example, the number of pesticides simultaneously found in urine in organic farmers compared to conventional. Well, this is basically what you would expect, but it is not always the case. So obviously we have to go back to questionnaires and actually the regional situations to do a good interpretation of the data. The second matrix we measured was feces. Feces would typically reflect exposure from the past 1 to maybe 3 days. I have to say that in many cases it reflects the unabsorbed fraction. So after oral intake, it has not been absorbed, it just is excreted as such. There are some exceptions, but a lot what we find has not been absorbed. The sample collection obviously is less straightforward, and it is hardly used as a matrix for human biomonitoring so far. The reason we included it was that it's much easier to cover a wide range of pesticides because we can look at the parent compounds, and for those we have the analytical standards available. The existing data hardly existent, so this is the first time this was done, certainly at this scale. And we covered almost 200 pesticides through multi-methods, and again, a dedicated method for glyphosate and AMPA. And here are the findings in these feces, a similar plot as I showed you before. In total, 190 pesticides were covered, and we detected 92 out of these. Some only a few times, others very frequently. Concentrations typically between 1 and 10 microgram per kilogram of feces, and glyphosate higher. Also here, a top 20 for the EU. Glyphosate was the most detected pesticide in feces in 70% of the EU samples, and in Argentina even higher in all the samples. Then we see a number of these old persistent ones that apparently are excreted to some extent through the feces. We all have them in our body, and there is some excretion through the feces. Now we have phthalimide, which is a metabolite of folpet, but may also have other origins, so a bit more difficult to interpret. And we have fipronil sulfone, so metabolite of fipronil, tetrametrin and permetrin, and those 3 compounds actually in the EU are not allowed to be used, are not approved for use as plant protection product. They're also hardly found in food, so there must be another reason, and our hypothesis is that this is originating from biocide use or use on pets and livestock. So it clearly shows that through human biomonitoring, we can observe exposure through other sources and other routes than diet alone. And also here we see for quite a number of pesticide differences in occurrence between Europe and Argentina. Glyphosate higher, fipronil sulfone higher, clopyrifos higher in Argentina, probably because it's still used or allowed there, and it has recently been banned in the EU. Also here mixtures, also in the feces, we find multiple pesticides per sample and the median number of pesticides. It kind of aligns with what we saw in urine. Again, Argentina and the Netherlands on the top, highest number of pesticides per sample, Denmark, Croatia lower. And also here we can see differences between the different subpopulations, again demonstrating that this is a useful tool to investigate this. One compound that has been mentioned a lot, and we have a lot of findings, so I also zooming in a bit on that here, glyphosate concentrations in feces, most frequently found in Argentina, also the highest concentrations, as we can see here, some difference between the European case study sites, Portugal and France, the higher than most of the others. Now, this is a bit of a complex slide, and it just combines the data we have for urine and for feces. And we see, for example, for glyphosate, that it's easier detected in feces than in urine. So this feces analysis really demonstrates we have exposure of almost all the entire population. We have detection of those pyrethroid metabolites higher frequently in urine, but those metabolites are non-specific, so it's difficult to tell from which pyrethroid, parent pyrethroid it comes. But with the data of the feces, we can say which one it was. So apparently cypermethrin, deltamethrin, and lambda-cyhalothrin, those are probably the most important parent pesticides where these biomarkers come from. Some of the pesticides we only find in urine, like the tebuconazole metabolite and 2,4-D, and others we find only in feces. So the combination of these two matrices provides us with a lot of detailed information on the actual internal exposure. That brings me to the take-home messages. The analysis of urine and feces reveals that humans are internally exposed to a wide range of pesticides, so not only through food residues, but also from water sources and other routes. Like what we saw in the environment, exposure to mixtures is the rule rather than the exception. And we can see that human biomonitoring can be used as a tool to reveal the exposure differences between environments and subpopulations. To be done, further monitoring to study exposure trends and also effectiveness of mitigation measures. And what is still an important part is further research on the interpretation of HBM data. So what we would like to see is guidance values to translate certain concentrations in urine, for example. When they are below a certain level, there is no risk. When they would be above a certain concentration in urine, there is a risk. And this is at this moment only available for a small number of pesticides, and this needs to be further increased. And with that, I'd like to thank all the collaborators on this and would like to thank you for your attention.
Thank you very much, Hans. Are there questions here? No. So online, do we have questions, Elia?
No, online there's nothing as well.
Okay, thank you very much. Thank you very much, Hans. So now we have the presentation of Maaike Gerritse from the University of Wageningen. So, Maaike, the floor is yours.
Yeah, we can see it, Maaike.
Thank you. In the past 2 years, I've been studying the effect that pesticides have on gut health, and I found that some pesticides influence gut microbiome composition, and some pesticides can negatively impact the cells lining the gut. So first, I'll explain a little bit more about them. Yes, so the gut microbiome, it consists of all the bacteria, fungi, microbes that all live in our gut, and we actually need them for our health. They help us balance our immune system, and they compete with pathogens. Because if the good bacteria live in our gut, there's no space for the pathogens to settle and to make us sick. But what happens if this gut microbiome is disturbed? Well, if there's a small disturbance, we can quite easily recover from that. But if there are disturbances over time, for example, you're taking antibiotics for a while, or alcohol can disturb the gut microbiome, and we might go to a dysbiotic state. And in this disturbed state, there are the composition of the bacteria is different, and this disturbed gut microbiome can actually contribute to disease. So you might have heard about the gut-brain axis. This describes how the gut microbiome actually contributes to our mental health and to our behavior. And this disturbed gut microbiome can contribute to psychiatric disorders, but also to neurodegenerative disorders like Parkinson's and Alzheimer's, but also metabolic disease, including diabetes and obesity, heart disease, lung disease, including asthma and skin disease. So now you can understand that in Sprint, we thought it was very important to research the effect pesticides can have on our gut microbiome. This is not done a lot. As Hans already explained, this is also the first time we have such a big data set on species in relationship with pesticides. And also EFSA is currently not using the gut microbiome in their regulation for approval of pesticides. So this is quite novel what we're doing here. And as Hans and Abdallah explained, we had species from about 700 participants in Sprint, and we were able to measure the different pesticide concentrations in this species. And from the same samples, we were able to determine the microbiome composition. And this microbiome composition in the feces, so the bacteria in the feces, are a very good representation of the bacteria that actually live in your gut. And with this data, we were able to ask the question, what is the association between the pesticide concentrations that we find in the feces and the gut microbiome? And we found that quite a couple of pesticides were actually associated with this gut microbiome composition. First, DDE-PP, which is a metabolite of DDT and hexachlorobenzene. These pesticides have both been banned worldwide for quite some time, but as you could see in the presentations before me, they are still found all around us and also in our bodies, where they do have an impact on our gut microbiome. Next to that, spirotetramate, an glucoside, is a metabolite of spirotetramate. This also had an effect on the gut microbiome, and pyrethroids and glyphosate also had an effect on our gut microbiome. These compounds, or at least some pyrethroids and glyphosate and spirotetramate, are still currently allowed for use in the European Union. Glyphosate and pyrethroids have been indicated in previous literature as well to have an impact on the gut microbiome. So our results here are in line with the earlier findings. Now, we also wanted to know what the effect is of the pesticides directly on the cells in our gut, on the human cells in our gut. Now, of course, we couldn't ask the people to donate a piece of their gut. That would be a little bit weird. So we set up a lab model to directly test the effect of pesticides on a model in the lab. This is an organoid model, which you can best understand by seeing it as a mini gut model. And this mini gut model was created from stem cells. Now, you may remember from biology, it might be long ago for some of you, that the gut has crypts and villi. And these crypts and villi, we have them so that we have a very big surface area to take up all the nutrients that we need from the food in our gut. And the stem cells of the gut, they reside in these crypts. So we were able to extract these stem cells from porcine gut and grow and proliferate them into this mini gut model. And these mini gut models, those organoids, they are quite an accurate representation of the actual gut. As you can see in the picture, they also have crypts and villi, and they contain multiple stems, multiple cell types, just like your actual gut. We exposed these models to 5 different pesticides: glyphosate, tebuconazole, cyprodenyl, fludeoxonil, and piperonyl butoxide. Piperonyl butoxide is actually a synergist, but as you saw in the previous presentations, it's all around us, and therefore it's also important to study the synergists and not only the actual compounds. active compounds. So we saw that the cells that we treated with pesticide showed increased stress and a reduced energy production. You may know that the mitochondria are the powerhouse of the cell. Well, the mitochondria in the cells that we treated, they were very stressed and therefore produced less energy. But of course, the cells need energy to fulfill their functions. So this is very bad. And we also saw an effect on the proteins involved in barrier functioning of the gut. Now, let me explain this barrier functioning a bit more. One of the important roles of the gut is to keep what is inside your gut and outside your body. We only want to take up the nutrients that we need, but we want to keep all the bacteria and indigestible food fibers. We want to keep all of that in the gut. And to do that, these cells anchor themselves very tightly against each other using special proteins. And if we have less of these proteins, if these proteins are impacted by pesticides, for example, we might go to a state that we call leaky gut. In this situation, bacteria or toxins from inside the gut might actually leak through these cells and enter the body, where they can cause a lot of inflammation, primarily in the gut area, but they can also travel through the body and cause inflammation in other places in the body. So it's very clear that we don't want this. The effects that I just described were actually the strongest for Figueiredo butoxide, which was the synergist, so not even the active compound that is usually the thing that we research the most. And the effects were also quite strong for fludeoxonil. But I want to stress that we saw effects for all 5 pesticides that we tested, including glyphosate, cyprodenil, and tebuconazole. We only tested 5 pesticides at this moment in time, and the effects of many other pesticides are still unknown. So this stresses that more research is definitely needed. And in Sprint, we're also focused on researching the effect of mixtures, because we hypothesized that if you have multiple of these pesticides at the same time, exposure to multiple pesticides at the same time, these effects might be stronger than if you're exposed to just a single pesticide. But as you saw before from the presentations of Hans and Abdallah, we're usually exposed to mixtures and not to single pesticides. So the take-home messages of this presentation is that we researched the effect of pesticides on the gut microbiome composition using field data in Sprint, and we found that glyphosate, spirotetramate, pyrethroids, DDT, and hexachlorobenzene can impact our health via modulating the composition of the gut microbiome. And we also studied in the lab the effect of pesticides on gut cells, and we found that Figueiredo butoxide, a synergist, fludeoxonil, glyphosate, cyprodenil, and tebuconazole can impact our health via increasing cell stress, altering energy production in the cells, and affecting proteins involved in barrier functioning. This raises concerns that warrant future research. For example, what other pesticides have the potential to impact gut health? What is the effect of mixture? And what is the effect of long-term exposure? Like we see all around us, you're exposed to very low concentrations on a daily basis. What happens in our gut when that happens? So I want to thank you for... It's stuck again. I want to thank you all for listening, and I want to thank also all the participants in Sprint and the people in Sprint, and my supervisors back at home in Wageningen. So thanks. Are there any questions?
Thank you very much, Maaike. Yes, we have a question here from Susanna. So can you please approach, because the microphone is not working well, and then like this, the persons online can also hear, please.
Thank you very much for a nice presentation. I just wanted to know, your study bystanders are those who apply the sprayers, pesticide sprayers. There are many high standards, like the family, the
We cannot understand you. Please, could you talk into the microphone?
The question was if we have included children in Sprint. For the field study, we did not include any children, but we did have an equal number of males and females. So I cannot say anything if the effect would be worse on the microbiome of children. But the microbiome of children is still developing, so this could be a concern indeed. For the lab experiments, it's a bit more difficult because we're using a model. So it's very difficult to model actually the difference between children and adults. Question.
Sprayers or those that
Could you please talk into the microphone for the lady who's asking? We cannot understand.
The question was who is included in the study. It was I used all the samples we have in Sprint, so it was a combination of the farmers, the neighbors of the farmers, and the consumers. And based on the concentration of a certain pesticide that they had in their feces, we saw that people with, for example, a higher concentration of DDT, which might be mostly farmers, but we did not make a distinction here. They had a significantly different microbiome from people with a lower concentration.
So I think the pesticide sprayers as such, if you do a particular study, then we know better because they are the most people who are exposed.
Yes, there was no significant difference between for the microbiome between farmers and the other. Yes, so the farmers, those were the people working with pesticides. Yeah, the other people.
So I thought
yes,
the second study only with them, and you can appreciate the understanding.
Yes, but there was no because we only have one time point, which is very difficult to say what the actual spraying event has as an impact, but there was no significant difference if we just look at farmers versus the other groups. But there was if we looked at the concentration of specific pesticides.
Thank you.
Thanks very much, Maaike. Do we have questions online?
Yeah, there's now one question from Jennifer that is asking if there is an accepted standard microbial profile which represents a healthy study participant. Should I repeat?
Yes, I'm trying to open the chat.
Yeah. So it asks if there is an accepted standard microbial profile which represents a healthy study participant.
No, I think this is one of the main things currently in the whole microbiome research that we would want to have. But the gut microbiome is a very personal thing, and just looking at a gut microbiome, it's very difficult to say this is a healthy person or this person has some health problems. So this is not something that we currently have. We do not currently exactly understand what makes a healthy microbiome and what makes a less healthy microbiome, but we do understand that it can contribute to many diseases if your microbiome is out of balance.
Okay, thank you, Maaike. We have another question here. Please, if you come from Declan.
Sorry, I'm late, of course, so I'm just at the tail end. But I just was looking at the last slide. I guess they were yesterday.
Yes.
Yeah. And really intrigued and fascinated that you raised the issue of microbiome and not being a specialist in anything, but I am aware that the microbiome is increasingly recognized as a critical health issue. including mental health, if I understand it correctly.
Yes.
And I'm therefore wondering, how can we, what is the message then to policymakers regarding this? I am aware of a range of microbiome research-related activities. I see that and read some of the literature, but how can we take them and communicate to the wider and particularly global, and by global, I was going to ask you if there's anything to say about the differences in incomes between advanced and less developed nations. So I suppose simply put, how can we advance this narrative to policymakers and in so doing improve health outcomes?
Yes, that's quite a complicated question.
Yes, sorry
Thank
about
you.
that. Yeah,
first, let me say that there is quite a bit of difference between the gut microbiome in more developed countries versus less developed countries. This may be because of a lot of different factors, but those factors can, do definitely include diet. In a more Western diet, we usually have less fiber, and that makes our gut bacteria less happy. So it's quite difficult to say if the effect of pesticides would be different between these different countries. But we do know that it's quite a big concern that in Western countries we actually lose diversity in the gut microbiome, and this is happening over the time of actually getting more modern and having a more Western diet. In terms of policy, as I said before, it's very difficult to define a healthy microbiome, but I do think we have to be very aware of all the things that can impact our gut microbiome. And as I showed, I think maybe right before you entered, there are some pesticides that do impact the gut microbiome. Yeah, I think it's important to take that into account when making a policy. And everything is multifactorial, of course, but it is a factor.
I think it's very important to include this alteration of the microbiome into the regulations. This is what we postulate. We need more sensitive indicators for pesticide regulation. And I think it's quite well known that there are relations between microbiome alteration and Parkinson's diseases, and these leakage gut syndrome, morbus Crohn, all these diseases are linked to alteration of gut microbiome. And I think it would be very important to include this into regulations, although we cannot say exactly which is the best combination in the gut, but we especially know that specific gut bacteria are correlated with specific diseases, and others are beneficial. So it would be very important to include this into regulation, and as well to make more help these researchers to get these in a very clear tool so that this can be applied on a global scale.
Also, just to complement the answer, in the last years that there are more intolerant persons to different aspects and to milk, to bread, and this is because there are not enzymes in the gut, and these enzymes are provided by the gut microbiome. So it would be also interesting to understand what is happening there and with the intolerance, because if we know the microbiome is affected by pesticides, then we are fomenting maybe more intolerant persons in our society related to food. But yeah, this is a comment.
Just one short.
Could you talk into the microphone, please? Because otherwise we cannot understand. And this is the last question because we need then a break.
Yeah, sorry to hold you. So as I understand it, and again, not speaking from a specialist standpoint, so the regulation of pesticides is advancing in the European Union and this is a good thing, I expect, you see. But my question, my concern is how is the European Union and its member states promoting a similar concern for pesticides in less developed nations? And partly the reason I ask this is because we have a lot of discussion during the Science Summit on data protection regulation and artificial intelligence regulation. And the European Union is promoting very, very, very strongly a global approach, global standards, mainly because in one view is that promotes European Union industry. So is the same approach being applied to pesticides regulation? So if Europe thinks a particular pesticide is damaging, is it promoting the regulation or the removal of that pesticide in poorer countries?
Yes, I think Violette can say something about this as well. I think what is very important, if we ban certain products in the European Union, we should also stop importing products that have been treated with those products. And this might also decrease the use outside of the European Union. And now I'll give the word to Violette.
Now, what it is this EFSA regulation is not reflecting real world situations. Very important aspects are not considered, as we can see here in the next session now. For example, we already saw atmospheric inhalation, atmospheric transport, sensible indicators. This is all not included into European regulations. Maybe the regulation of Europe is better than in other countries or continents. I can't judge this, but it is far away from assessing real risks that people are in our environment is exposed in the real world. And we really need an improvement and a real world risk assessment and a transition to more sustainable agriculture. That is very, very clear. And I would not suggest to convert the EFSA system now globally, because there's a lot to do to really come up with a real world assessment of the risk of pesticides to humans and the environment. But now I would like to ask to make a maybe 5 minutes break only, so that we then can continue. Or what do you think, Esperanza?
Yeah, thank you very much for this great discussion. Thank you very much, Maaike. Thank you very much, Declan. So we have 5 minutes break, please. So now is yes, we see each other in 5 minutes, please. Just a small break. 5 minutes. 5 minutes break. Coffee break.
Yeah.
5 minutes, please.
Esperanza, could you just call Maaike, so maybe she could quickly answer just for so others can go to the break, of course, but there's a quick follow-up question in the chat that might be good.
Here. Yes.
Yeah, sorry, because for those online anyway, this 5 minute break doesn't matter so much. There's a follow-up question from Jennifer together with another person, that when you said that you only know when it is out of balance, then what is a microbiome out of balance? Considering what you said about the standard microbial profile.
Yes, that's also a very complicated question, because again, it's very personal, but there are specific bacteria where we know that they are beneficial, and in this biotic state you might have less of these bacteria and you might have more pathogenic bacteria. So that's in general what I mean by that.
Okay, so anyway, there for what could be considered a healthy person, there's a standard typical type of bacteria you expect to find, and that's how you judge out of balance in the presence of pesticides that you know about, at least, correct?
Yes, but again, it's very personal and very complicated to say just based on a microbiome. You can't say if a person is healthy or unhealthy, but you can say if a person is sick, usually the microbiome is also different from healthy people.
Okay,
so the
so
other
what
way
you
around
would consider healthy people. Yeah.
Yeah.
Thank you very much.
Thank you. Yes, I would like to talk about the pesticide exposure by air to agricultural sites in Europe that we took from the Sprint project. And yeah, I'm a PhD student at the Helmholtz Center Herion in Germany. And first of all, I would like to give some background information. So why do we analyze pesticides in the air? So when the pesticides applied on the field, they can end up in the atmosphere during the spraying by spray drift, or also after spraying by volatilization or wind erosion. And then depending on their properties and on the application method, they can partition in the air. And then they can be transported through the atmosphere to non-target areas, and can there also be deposited by wet or dry deposition and can affect these areas. And so what we did was that from the sampling sites within the Sprint project, we took 2 sampling sites in the Netherlands and in Portugal. And at each site, we installed a high volume air sampler, with which we sampled for a duration of 1 to 2 weeks per sample for a total period of 14 months. So we sampled in 2021 and 2022. And the sites that we chose represent different climate zones in Europe and different farming practices. So in the Netherlands, there were potatoes and sugar beets grown during the sampling period. And in Portugal, it was an area where wine was grown. And the sites were also chosen because for the high volume sampler, we need a power supply. So this was also an important aspect when we decided which sites to choose. And what we did is that we sampled the particulate and the gaseous air phase separately. So this is important because for the pesticides that are present in the gaseous phase, they can be taken in by inhalation. And for the compounds, for the pesticides bound to the particulate phase, it depends on the particle size if they could end up in the respiratory system. And so when we look at the results, we analyzed a total of more than 300 pesticides in all these samples. And in total, we detected 96 pesticides and pesticides metabolites in concentrations between 1.5 and 10,000 picogram per cubic meter. And when you look at the figure on the right, you can see this is the total pesticide concentration per sample in picogram per cubic meter, and it is the sum from the Netherlands and Portugal. So all samples were taken into account. And as you can see, the concentrations per sample were for 50% of the samples between 100 and 1000 picogram per cubic meter. And when we then look at the number of detected pesticides within the samples, you can see that for the Netherlands, more pesticides were detected in both the particulate as well as the gaseous phase compared to Portugal. And when you look at the phases separately, you can see that it was in each country quite a similar number of pesticides detected in both phases, although a little bit more pesticides were detected in the particulate phase. And when we then have a look at the temporal variation of the pesticide concentrations within the sampling period, you can see here 2 graphs. The upper one is from Portugal and the one on the bottom is from the Netherlands. And on the x-axis, you can see the sampling period. And on the y-axis, you see the total pesticide concentration per sample. And this is the sum of particulate and gaseous phase. And as you can see, the concentration in the Netherlands was higher than compared to Portugal in the samples. And you can also see for both countries that the concentrations were higher during the spring and summer months for both years. And this is also correlates with the application of pesticides. So this was also during the growing season when pesticides were applied. And when we now look deeper into some examples of pesticides that were detected, here are some 2 pesticides. It's both fungicides that were detected at the sampling site in the Netherlands and that were also applied on the fields that were located around the sampler. And as you can see, these were also mainly detected in the spring and summer months. And when we look at the application of these compounds, you can see that fenpropidine was applied 2 times and mandipropamide was applied 5 times during the spring and summer months. And when the pesticides are applied, the concentration goes up, and then you can see that it slowly decreases after the application. And yeah, when we now have a look at the estimated maximum human exposure that can be caused by inhalation, you can see here again fenpropidine on the left, the compound that you also saw before. And this is the compound that was detected in the highest concentration in all the samples that we took. So that's why it's the maximum human exposure, because it's the highest concentration that was detected. And on the right, you can see the sum of the total pesticides detected per sample. So this is also the highest concentration of pesticides that were detected in one sample. And the values of the daily inhalation rate were calculated by using the expected inhalation volume and body weight for adults, children, and infants. And as you can see, for infants, the daily inhalation rate is higher than for adults. And you can see that for all these values range between 3 and 17 nanogram per kilogram body weight per day. But it is hard to put these values into perspective because we don't have any thresholds for airborne pesticides in Europe or in total, because it's the same. And as Violette also said before, there's no EFSA regulation that includes the atmospheric transport. So there are no thresholds available where we could see, could these concentrations or this daily inhalation rate cause an effect on humans. But as we have quantitative results, so you have a concentration of pesticides in the air, this is the first step for exposure assessment, and then also data that can be used for risk assessment. And when we now also look at the hazardous properties of the airborne pesticides that were detected here, these are the pesticides that are detected at both reference sites, so the total number of pesticides. And you can see here the same properties that Violette also used in her slides. And as you can see, especially for compounds that, can cause reproduction effects or can be carcinogen, you can see that quite a lot are, it is yes or possibly carcinogen or can cause reproduction effects. And also for the others, so mutagen, neurotoxicant, and endocrine disruptor, there are quite some compounds that that have these hazardous properties. And what is also important to be noted is that for most of them, more than 20%, and for the endocrine disruptor, even more than 50%, there's no data available for these pesticides. And so for these compounds, it's not possible to say, can they have a hazardous effect, or can they cause some issues to humans or the environment? And so, to conclude these results, it is important to know that the atmospheric transport is an important pathway for the exposure to pesticides. So we can measure them in the air. We also measure pesticides that were not applied on the field. So they must be transported from other areas. And also the quantitative data for gaseous and particle-bound pesticides is a really important part to do transport modeling and risk assessment. So we need this quantitative data in the air to do this. And then also what is needed are toxicology-based thresholds for individual pesticides in air. So this is what Violette said, it has to be included in the EFSA regulations so that these values that we have now can be put into perspective. Yes, and with that, I'd like to thank you, and I'm happy to answer any questions.
Thank you very much, Freya. So do we have questions here? No? Online? Do we have questions online?
Yeah, there's one question from Andrea Pisotti. If pesticides do stick to particulate matter?
And they do.
Yeah, and yes.
So the particle-bound pesticides, they stick to the particles and then can be analyzed.
Okay, so thank you very much, Freya. So now we go to the presentation of Dirk Goossens. So please, Dirk, the floor is yours.
Okay, well, welcome everyone. My name is Dirk Goossens from Wageningen University and also from the University of Leuven in Belgium. And you can see my co-authors here who are mainly also from Wageningen University, Celia Bento, Margus Rezay, Bart van Straten and Rima Osman. And there is also Freya from Germany who gave the previous presentation. And the title of my presentation today is Pesticide Transport by Wind in Sediment Originating from Agricultural Fields. Just a quick reminder, I already also said this. Airborne transport of pesticides can occur via various processes. You have liquid drift, which means that spray droplets containing pesticides are transported by wind. The second process is vapor drift. This means that pesticide molecules volatilize and are then subsequently transported by wind. And then you have the third process, particle drift. This means that pesticides are bound to soil particles that then are released into the atmosphere and further transported by wind. And my presentation today will be mainly on the third topic, particle drift. Why can particles be released? This can be done by means of various processes. That can be natural processes. The most obvious one, of course, is wind erosion. And also human activities can release particles into the atmosphere. For example, when the farmer is tilling his field. You already have seen this, I think. If farmers are driving over the field, they are producing huge dust clouds. And also off-road driving, which means that if people are driving over unpaved surfaces also, then lots of particles are released into the atmosphere. Now, what I'm going to present in this talk is showing you a few results that have been obtained during a field experiment that we have conducted in 2022 in the northeastern part of the Netherlands. It's the same site as the one that Freya commented upon in her presentation. You can see here a satellite image of the area. You can see the agricultural fields. Just to give you an indication, they are very rectangular and the size of each field is approximately 5 hectares. Here you have an idea of how the landscape looks like, and it is not unimportant. You can see that there are almost no topographic obstacles, and the land is very flat. It's a very open landscape, which means that the wind can reach quite high velocities here. And in addition, the soil itself is very vulnerable to wind erosion and also to dust production when the farmer is working on it. So it's an ideal place to check particle drift and pesticide transport by particle drift. Now, if you want to study this, then of course you have to collect your particles. How did we do this? I can show you here. For some reason, I don't know why this doesn't work. Okay, here I can see the next slide. Here you can see how we did this. You see the experimental setup. It's a very simple one, but nevertheless a very efficient one. So we installed several small towers. You can see 4 of them here. We had more, but you can see 4 of them on this slide. We installed these towers near the border of an experimental field. And to the towers, you can see these yellow samplers, which are orientating themselves into the wind by means of those wind vanes. And we know the efficiency of these samplers, so that means that we can collect the samples and also determine the exact quantity. Some technical information about the experiment. It took approximately 2 months, 8 weeks in total, and we collected the sediment after every 2 weeks. Why 2 weeks? Because we need, of course, a minimum amount of sample to get it analyzed for pesticides. So we had 4 different periods of 2 weeks. All the sediment was analyzed by Freya, and she analyzed it for a total of 208 pesticides. Which one did we find? In all 4 periods, there was a lot of glyphosate and AMPA. And this was not a coincidence because by accident, we were very lucky, the farmer decided to spray his field with glyphosate 1 day after the measurement started. So this gives us a very nice opportunity to follow how glyphosate and AMPA behaved during our experiment. And in one of the periods, we also found 6 other pesticides, of which you can see the names here. This presentation will be mainly on glyphosate and AMPA for the reason that I already explained to you. What did we measure? First of all, we looked at the presence of glyphosate and AMPA in the air, so the airborne concentration. The upper 2 figures here show you, left for glyphosate and right for AMPA, the airborne concentrations as a function of height. And then you can see, be careful because the x-axis is a logarithmic scale, be careful of that. It shows you that the most glyphosate and AMPA was transported quite close to the ground. So let's say in the lowermost meter of the atmosphere. The 2 lowermost pictures show the concentration of glyphosate and AMPA over time. And 0 in the x-axis means the day that the farmer sprayed his field. And then you can see that in the first, approximately 6 weeks, which is almost 1.5 months, the concentrations remained quite high, much higher than I had expected. So airborne concentration is one thing, but of course we are also interested in the transport, and therefore I calculated the transport flux, both for glyphosate and AMPA, again, as a function of height for the 2 upper figures, and over time in the lower 2 figures. And you can see that these pictures are almost the same, and they are quite similar to what we already saw in the previous slide. So we knew that there was an important transport of pesticides. And then, of course, the question, but then there are 2 questions. First of all, how far will these pesticides be transported before they settle down? And the second question is, where exactly did the wind take them to? So which were the areas that were later affected? To answer the first question, I remind you that we are here talking about pesticide transport by particle drift. So the distance the pesticides will travel will be determined by the distance the particles to which they are attached will travel. And then you have to take into account the different aeolian transport modes that exist. And those that are of concern here are situated here left. I suppose that most of you will be familiar with these terms, but if not, just a quick explanation. Saltation means that particles are transported close to the surface in the lowermost centimeters or decimeters, not more. And they will settle down rather quickly. So their transportation distance is normally of the order of a few meters to a few tens of meters. So they will stay rather close to the source. Modified saltation means that these particles can go to heights of several meters, up to 10, 15, maximum, I would say 20 meters. And they can be transported over distances of a few hundreds of meters to 2-3 kilometers. This is not the case for suspension, because when particles are transported in suspension, they are floating in the air, and their transport distances are much larger. For short-term suspension, we are talking of a few kilometers to a few tens of kilometers. And for long-term suspension, we are even talking of a few tens of kilometers to over 100, 200, and for the finest particles, even over 300 kilometers and maybe even more. Now, to check what the situation was during our experiments, I calculated the proportion of these different transport modes for glyphosate and for AMPA. And I also included the PM10 fraction, which is a fraction less than 10 micrometers, because this is the fraction that is of concern when it comes to inhalation into the human respiratory system. And you can see that both for glyphosate on the left and for AMPA on the right, that most of these products were transported during our experiment in short-term suspension. If you then add long-term suspension to it, then you arrive at a percentage of over 50%. So this means that more than half of the glyphosate and AMPA that was present in the air at our site during the measurements had the capability of being transported over very long distances. We are talking here of tens of kilometers and even more. So they can, when they settle down, affect large areas. Then for the second question, where exactly were they transported to? To answer that question, we have asked our colleague of the meteorology group at Wageningen University to apply or to check it with a dispersion model. And I got the results until only quite recently. I got them 2 days ago, so they were just in time to show you. What you see here is the transport trajectory. I don't know if you can see my cursor, but the starting point here is our test field, and that's the source. And you can see the transport trajectory. You can see it for 3 different particle size classes. The one here represents short-term suspension, and these 2 on the left, represent long-term suspension. And well, the arrow indicates the wind direction, and we have 2 snapshots here, 2 time snapshots, and you can see them here. It was on the same day, but for the first 3 figures here, they show what happened during the day hours. And the 3 figures below show the situation in the late evening, and which is also representative for the night. What you can see is that during the day hours, so the upper figures, the transport patterns were quite comparable for these different size fractions, but not so in the late evening and night. And you can see, for example, that at night, there was still a significant transport of the finest particles, but not of the coarser particles. And look at the figure in the bottom right. This is for short-term suspension, but there was almost no short-term suspension at night. This is not, of course, a coincidence, because normally during the day hours, wind speeds are higher than at night, and also the degree of turbulence is much higher. So that means that at night, coarse particles will have difficulties to stay aloft for a long time. And so that's one thing that I can show you here. A second result of the modeling, and this is also quite interesting and also important, what you see here is the start of a movie. I will let it run in a while. So the 3 pictures above again show the transport trajectories for the 3 same particle size classes. And the 3 figures below, they show deposition. And so the model also calculated deposition. Now I will start the movie. It takes a few seconds, and it represents what happens on the 9th of May. It started around 3 o'clock in the morning, and it ran, I think, until 11 or so in the evening. So let's see what happens if wind direction changes. I hope I can start the movie. Okay. There it starts. So we are still in the morning hours. The wind is blowing from the east. But as you can see, the wind direction changes during the day. We are now shortly after noon. The wind has turned to the south, and then it's turning to the southwest. And in the evening hours, it turns to the west, and at night, it made a whole turn. And look now at the deposition pattern. The figures on the, especially the figures on the left below, you can see that now the whole area around the source has been infected. So this means that a changing wind can be very important. And this shows that particle transport by wind and also pesticide transport by wind is something that is important because it will affect huge areas depending on whether the wind direction will change or not. And normally it does change during the day, and it does change during a week, and it does change each time that you select a period, the wind direction will change. And this is an important difference if you consider particle and pesticide transport by wind compared to pesticide transport by water, because water flows in principle only in one direction. It flows downstream, but not so for wind. Wind can flow from any direction, and therefore pesticides that are carried by wind can be dispersed everywhere. Now, this also means that it is possible, and Freya already told you about this, it is possible that some of the pesticides that we detected were not applied locally. Probably they have been applied, I don't know where, upwind, probably very far away from where we measured them. And indeed, if we look at the 6 other pesticides that we detected, and we detected them, because you can see their concentrations here, we checked with the farmers, and none of these 6 pesticides were applied locally. Not in the field itself, and also not in the wide surroundings during the period where we measured them. So this shows that either they should have been applied elsewhere, far upstream, or the alternative, of course, is that they have been applied locally, but then long ago, and that their degradation was so slow that perhaps they were applied last year, 2 years ago, and if the degradation is quite low, then they are still present, partly in the soil, and if then the farmer is tilling his field, or if then there is wind erosion, these old remnants, so to say, will then be brought into the air. And this brings me to my final slide, which is the core, or which are the 2 core messages of my presentation. One should realize that the spread of pesticides by airborne transport of soil particles definitely, and already Freya said this, it is a very important process that can affect large areas, even areas that are located at great distances from the source. And as a consequence, and it is already what Violette said in her presentation, EFSA fate modeling should urgently include pesticide residues in airborne dust. So these are the 2 core messages of my presentation, and I will stop here. Thank you for listening. And if there are any questions, I will be happy to answer them.
Okay, thank you, Dirk. Thank you very much for your presentation. We have time for one question, only one question. Okay, so we have the question here. So, yeah, so please.
Thank you very much for a very informative presentation. My question is, we have started using drones for pesticide spraying, especially in developing countries like India. So that may help the sprayers, but what about the airborne spray? Can you just tell me whether drone should be used or not?
Can you repeat the question, because I didn't hear it quite well? Perhaps Esperanza, if you could repeat the question.
Yes, whether drone applications, if they apply pesticides by drones, this will help to reduce the airborne transport, yes or no?
Exactly, yes. That was the question.
Help the
I have the idea that it will even become worse, because drones are not flying very close to the ground. So that means if they are flying in the air, and there will always be some wind speed. So that means that if they are spraying with drones, I think that the chance that the fine droplets will reach the surface, the chance will be low. So I think it's better that the farmer uses his classic system instead of using drones, unless, of course, he's able to get the drones very close to the surface.
Okay, thank you. I think, Peter, there's a little bit of time issues here.
Okay.
So,
okay, so thank you very much, Dirk. Thank you also for the question. Now we go to Peter Fantke from the university, from the Technical University of Denmark. So Peter, just a moment. Can Kayode is doing. Yeah, perfect. So now the floor is yours.
Thank you so much. Welcome everyone. Good morning, good afternoon, wherever you are. Let me share my screen and let me know when you see it.
Yeah, we can see it. Yes, you can see it.
Okay, perfect. Then I will start right away. Thanks for giving me the floor. I'm really honored to be part of the presentation team here today from the Sprint project. My presentation will be about going beyond what we can measure in the fields, in humans, in ecosystems, in organisms, and everywhere else, and trying to puzzle the different pieces together to estimate from all the measured data with underlying models, puzzling the pieces together into estimating what are possible environmental ecological impacts of pesticide use over their entire life cycles, not just looking at the pesticides applied on the field, but also considering the supply chain across the different production systems. I have collaborated on this work together with my colleagues Farshad Sohedifad and Yu Zheng. Let me see whether I can move on. Here we go. So the approach in one slide, in a nutshell, is we have looked at two different aspects. We have looked into we have developed a framework for assessing direct impacts of the pesticides applied in the different farms across different countries. For that, we connected pesticide application data that were derived from questionnaires from farms, as others have presented, with models for estimating emissions as a function of what crop stage and what application method has been reported. And further on, these emissions have been coupled with integrated fate exposure and effect models to estimate how these pesticides distribute in the different environmental compartments, how they expose ecological receptors and humans, and combine that with different effects based on available effect data. In addition to that, we have in the bottom of the slide, you can see we have assessed the supply chain impacts of the different pesticides that have been applied in the three systems, like conventional farming, IPM, and organic farming. Here, we have mainly collected and relied on reported supply chain data, for example, information on energy consumption for machinery use for applying pesticides or for synthesizing pesticides. We have then coupled the whole data system with the life cycle assessment framework, where we, on a consistent unit to compare the different farming systems, could make a big inventory of what are the data that the systems use, like leading to emissions into the environment, greenhouse gases from energy consumption, water use for resource consumption, for example, with different impact categories on climate change impacts, on human toxicity, on water use, on eutrophication, and so forth. So these two contrasting frameworks have been applied to see which of the two, the direct impacts of pesticides on the field applied versus their supply chain impacts, are actually driving overall potential impacts of pesticide use. This is the main result for the farm level impacts here. So we see mainly the dark blue across the different human health, ecosystem quality, and natural resource protection areas. Then we look at the different farming systems, conventional, integrated pest management, and organic farming. So we have always three per area we want to protect. We see mainly the dark blue pillars are dominating, especially for human health and ecosystem quality. And the dark blue bar is actually associated with the direct impact of the pesticides applied on the fields. That means when we translate impacts of pesticides on humans via the different exposure pathways and routes, of which some other presenters have already discussed, that will dominate as compared to impacts from energy consumption for the supply chain of the farming pest control systems on human health. And it is even more relevant for the ecosystems, of course, less so for the resources consumption, where especially the machinery use here on the field is a big driver, but also the production systems of pesticides. So in a nutshell, what we learned, the first thing is direct impacts of pest control options are really dominated by the in-field or down the drain impacts from using the pesticides. Now we have put these farm level data into the wider context of how would it look like in terms of impacts on humans and ecosystems when we scale up these farm level information to national level. So we focused here on several European countries for which we had farm-level data from the SPRINT project, and looked at the pesticides used in this project and applied them via a global database that we could receive from an external data provider. We combined these pesticides with all the crops reported in the global data set. So beyond what the farms reported, because of course, farms only represent a very small fraction of the overall use of pesticides, and of course, also in terms of which crops are included in these farms versus national level. So we have gridded these pesticide national level data coming from the global data set and projected them on a spatial data set on the crops produced in the European countries considered. So this was a 5x5 argmin data set for each crop, and different pesticides we projected directly on each crop separately. So we took an average across 5 years for the pesticide data between 2016 and 2020 to get a stable average mass applied per hectare estimate. We used these data sets then, which are specialized pesticide applied data estimates, with our emission models to distribute these applied pesticides on the environment, partly going to air via wind drift, partly going to water and soil via deposition, and directly on the field crop, again via deposition. So what we see here is a first glance on the purple countries, which are the considered ones for the SPRINT project, although we also have data for the green countries here, but we don't focus on them now. But you can see, a spatial pattern of a mass applied per hectare and over the whole year that really spans over 5 orders of magnitude. So it really matters which crops are grown and which pesticides are applied. And here we just see the example of the most widely used chemical across countries, which is glyphosate. And we see substantial differences in spatial application pattern, of course, showing that it is very important to make local differences when assessing these. Now, going from these emissions into an environmental fate system, estimating then concentrations after the cumulative mass applied of these pesticides over the whole year would give us, again, a spatial distribution of concentrations estimates, again, spanning over 5 orders of magnitude. And what we can see here, our highest concentrations in the dark brown ranges here in, for example, Portugal, northern Italy for glyphosate, we can see we reach concentrations that are in the range of low to medium measured concentrations also, which is up to 0.5 milligram per kilo dry weight of a soil. So even though we have many assumptions behind these emission and fate and exposure models, we come to reliable orders of magnitude that are comparable with what colleagues actually in other universities can measure in the fields at local scale. Now, we have the same thing for freshwater, where we reach as highest concentrations up to 0.03 milligram per liter water, with a factor 10 to 20 uncertainty around that, which is still substantial on the high end of the concentration, still spanning, again, more than 5 orders of magnitude just for one chemical, as a function of how it is applied, on what crop is it applied, and when is it applied. Now, when we go beyond one chemical, for which you have seen several maps here, and scale it up to all the 120 pesticides that we have considered across the different farms, but across all the crops grown in these countries where the pesticides are applied to, we find here cumulatively the total impact on human toxicity, where we considered mainly the residues in the food crops as driving environmental pathway for human toxicity towards pesticide use. And we considered both cancer and non-cancer toxicity effects extrapolated from animal data. So yes, there is, of course, uncertainty involved here. And countries cannot be directly compared, of course, because different countries considered different crops. So we have to be careful that we blame one country growing a certain crop, which might not be able to be grown in another country due to environmental parameters. However, summing up the total human disease burden for the pesticide use considered here over 1 year gives us roughly 2 million lost healthy life years across the European considered population, which could be translated into 28,000 roughly statistical human lives lost. So this is not persons dying from pesticide, but if you look at several 100 million people in the European countries considered, then we would have a certain small risk across the population that cumulatively would give statistical human lives lost. So this is not specific people dying here. However, mainly non-cancer effects are really driving the overall story. And we can see here with the dot, the contribution of cancer effects is really beyond a percent, often below a percent usually. So the blue dot is just on the right side scale, the contribution of the cancer, whereas the orange bars means the actual total lifetime loss cumulatively in Europe for the 1 year pesticide use per country where these pesticides have been applied. Now, what is important is to see that only a handful of pesticides drives more than 80% of the total human impacts. And we will see similar pictures also for the ecological burden. So we have incentives, even though all these estimates are really based on modeling and data, we will see that beyond uncertainty considerations here, that still only very few pesticides drive overall impacts in Europe and certainly also in other regions, which gives us good incentives for targeting priority chemicals for substitution. A similar pattern for ecological impacts here, we see on the blue side freshwater impacts, on the green is natural soil impacts, the brown is the agricultural soil impacts, and then the dot is really the total. So overall, we have, of course, highest impacts here in France and Spain and Italy as our European vegetable and fruit producers. And these impacts would not be different if these fruits and vegetables would be grown in other countries. So this is really about optimizing the systems in terms of pesticide use that should be reduced beyond where the pesticides and the crops are actually grown. Again, we have a total impact from the selected pesticides used over 1 year that cumulatively gives statistical 80 million potentially disappeared fractions over all the square meters considered in Europe, in these countries, over 1 year. And that would be, of course, hard to translate into one species, two species actually dying, but it shows we have an actual reduction across species that is substantial due to pesticide use. And that is, of course, not something that is very new. Again, most important, we have 3 pesticides driving or accounting for 3/4 of the total impacts on ecosystems in the considered countries. And we have not considered all pesticides even here. Final slide before the conclusions is now, can we translate these impacts? Because the whole exercise is about how do we manage to reduce pesticide use? How do we account for and benchmark it against market levels for selling the crops that we are producing and use pesticides on? And we translated the human impacts and ecological impacts into societal costs that society will have to have if we have, for example, pesticide health impacts bringing to hospitals, or if we have some ecological damage on our environment and cannot deliver any ecosystem services anymore. If we translate that, our impacts from the considered pesticides over 1 year here in Europe into cost, we would arrive at a substantial amount of, our estimate is 150 billion euros for the 1 year pesticide use across all these 10 countries considered. That is substantial, and even though people might argue there might be a factor 100 or 1,000 uncertainty around that, that might be true, but even if we apply 3 orders of magnitude uncertainty here on the high end, if we really have uncertain data, we still go in the hundreds of millions euro per year of pesticide use that we arrive at societal cost level. The main take-home messages from that exercise is that for the broader European population, residues in food crops from pesticides dominate population exposure. For residents closer to fields, of course, we have seen earlier, it is more dust-related ingestion, for example. Chemical pesticide use needs to be reduced across all farming systems, including organic farming, where no xenobiotic chemicals are used, but still copper as very toxic fungicides are used, and accounting for a lot of the disease burden environmentally and human-wise here. And the third point is substantial market external cost of pesticide use on humans and the environment make current pest control really unsustainable. So we really have to do some action. Thank you.
Thank you very much, Peter. So are there questions here?
I imagine.
No? Online? Are there questions online? Celia?
Online, well, there's just one question, but I don't know if it's specifically for Peter, asking if the data on the Sprint project results is going to be presented to be considered on the renewal of glyphosate coming soon in the EU. This is more for you, Violette, actually.
Yeah, we hope that this week or next week, our manuscript of the overview manuscript, which is related to my presentation in the beginning, will be published. We already answered the comments of the reviewers. We resubmitted it, so we are hoping every day that it will be accepted, and we hope that it will be accepted very soon. Yeah. So we hope that this will be done soon. And Peter as well, I think you are quite far in preparing manuscripts on this, isn't it?
Yes.
So there was a question as well, and this has to be discussed and afterwards whether this session is recorded. And the question was whether people can get access to the recording session. So we will ask each individual presenter whether he or she agrees that this will be his recording or her recording will be distributed. Yeah, we will do this after the session. Okay.
Okay, thank you very much, Peter. So we now have the presentation of Kayode Jegede from the University of Wageningen. So Kayode, the floor is yours.
All right, so I think I'll just continue because of time. So first off, I want to thank everyone for being very patient so far. I'm one of the conveners, I'm the co-convener, with Violette being the convener. So we have one more presentation after this and then the final presentation. So please don't go and listen for us. So I hope we've learned one or two things from our presentation so far. Now my presentation is on pesticide risk in terms of the mixtures on the terrestrial ecosystem. So for me, more than 25% of the world's biodiversity lives in the soil. So I'm not concerned about what happens with biodiversity in the soil. And as well, pollinators are responsible for about 30% of global crop production in the world, where bees take up about 70% of that. So my concern is about the soil. So soil functions can be impaired. And in some parlance, we usually say soil is a finite resource because once the function is impaired, you may not be able to get it back to work again. Then you've wasted the soil. For some people, they just see that soil is abundant everywhere, but for us as soil scientists, we count it as being finite. So we have to take care of the soil. So one of the ways by which soils can be disturbed or polluted is by pesticides. So, and we all know pesticides are by nature harmful. That's the design, of course, to target species. But in this case, a lot more studies have shown that non-targets are often also affected. And that's why we usually do risk assessment of pesticides. So my next slide. So pesticide risk assessment. So this is just want to illustrate how we do pesticide risk assessment. But it's okay. So one thing I wanted to say about this is that pesticide risk assessment currently is being done using single pesticide data. So for example, risk assessors will take maybe glyphosate and say, okay, glyphosate is safe at this concentration. And they do that with tests on test species, very few test species, maybe 1 or 2 or 3 standard species. But then mixtures in reality, in the environment, pesticides exist as mixtures. I'm sure we've all learned 1 or 2 things from all the presentations so far up till now, that pesticides exist as mixtures. So it means that a risk assessment should be done based on mixtures. So, and that's what Sprint is about, using different mixtures of pesticides and as well as more than 1, 2, 3 test species, and going beyond the normal standard species that we use. And with that, we are sure to get a better risk assessment to protect the environment. All right, very quickly. So these are the biodiversity in Sprint. So we use the springtail, Posomia candida, which is one of the most abundant microarthropods in soil. And they are involved in nutrient cycling and organic matter degradation. And as well, we all know earthworms. So earthworms they usually call them farmer's friend. So we use them as part of the test species, and as well as the honeybee that we all know, Apis mellifera. All right, very quickly, so these are the setup of the lab, just few pictures here and there. So figure 1 shows how the experiments, the earthworm experiments are conducted in the lab and under some constant conditions there. And then figure 2 shows how they extract the earthworms after maybe 56, 74 days of experiment. We extract the juveniles out of the soil using water bath. And then figure 3 shows like a grid of showing a grid of how we can estimate the number of bees, for studies. Figure 4 shows the springtail under constant conditions in an incubator. And the concentrations we use for our study, because it's an ecotoxicology study, we use the measured environmental concentration. So based on the Sprint project, so we used the concentrations measured in the environment, and we tested them in the lab to see the effect on the organism. We also use the predicted environmental concentration, which is a modeled concentration by EFSA. And then we try to look at, in case there's a spill where we have high concentration of pesticides, spill, whatever, we have to look at the 5 times the predicted environmental concentration to see what the effect can be on these organisms. So very quickly, now to the results. So pesticide initial effect. One thing I want you to see here is that if you look at all these lines show the threshold values where we have effect. But then these dots and these signs show the concentrations we use in the lab. So for all the pesticides in this particular mixture, all of the concentrations we use were below the threshold value where we expect effect. Therefore, for this study, we're expecting no toxicity because the concentrations we used were very low compared to what will cause toxicity for each of the pesticides. However, if you look at the left downward panel where we have the graph, there was a significant toxic effect of mixtures. So if we consider just single pesticides, for example, you may say glyphosate is safe, but then if you consider glyphosate, AMPA, and other pesticides together, then you find effect. And that's exactly what we found here. So we can see that there's significant effect on the reproduction. That's about less than 50% reproduction was gotten. So which means that there was an effect of pesticides mixtures. And also on the springtail, the same story like the earthworm. So all these concentrations, they were far above, the threshold value were far above the concentrations we used. So we're expecting no effect here. However, if you go down to this, you can see significant toxic effect, where more than 50% mortality was recorded in this study and almost 100% inhibition of reproduction. So which means that pesticide mixture, it's very important that we check that. Also for the bees, these are preliminary studies, so we don't have the data yet, but this is just a visual clue to see how pesticide mixtures can affect bees. So for the first upper panel where you have no pesticides, you can see that all the grids are covered with bees, which signifies a healthy colony of bees because they are all over the place. But if you look at where the pesticide mixtures are, you can see a lot of empty grids, which means that they've been affected. So that's a shrinking colony of bees. All right, so the next thing is the risk detection. So why did I emphasize, or why are we emphasizing using more than one species for tests? So this is exactly the reason. So if you look at this, so if you use just one species as a test, just like we do normally in risk assessment currently, then out of the 11 mixtures we tested, we can only detect risk for 5. That's using just one species, the standard species. But then if you increase it by 2, if you use another, if you increase the number of species used or tested, we were able to detect more, we're able to detect risk in more mixtures. So we're able to detect 8 out of the 11, which means based on these 3 mixtures out of the 11 are safe, while 8 are not. But then if we increase that further, using all the 3 species here, we have all the 11 mixtures being risky. So which means that that's why we're emphasizing that risk assessment should be based on biodiversity, based on more than one species, and especially native species. So that takes me to my discussion. So what am I trying to say so far? Mixtures are not safe at supposed safe levels for single pesticides. And when we increase the number of pesticides, we can increase the detection of our risks. Therefore, we're able to protect the environment better. So what do we do going forward? We've established that pesticides can be dangerous to terrestrial biodiversity. And then the current pesticide risk assessment method should be revised. So EFSA, for example, they consider single pesticides, but then based on this study, we are saying mixtures of pesticides should be considered in risk assessment. And then that is when we can be more confident with whatever data we're using to protect the environment. And then focus should be on biodiversity, not just single species, not just standard species like we do. So now we only have maybe one standard species for earthworm, which is Yersinia foetida or Ambre, and then springtail, one standard species. But in our case, we are advocating for more species, especially native species, so that with that we can detect risk better and then protect the environment. So this will be going forward. This will be what risk assessors or policymakers should be looking at, how they can change, how they can modify risk to be better than it used to be. So with that, I welcome your questions. Thank you for listening. Sorry for all the logistic problems. Thank you very much.
Okay, thank you very much, Kayode. Do we have questions here in the front? Yes, please. But can you approach, because our microphone is not working, and like this, the persons online can hear you also. Sorry for this. So,
actions that you have for this, once you have this, how do you mitigate?
Can you please approach the microphone, so otherwise we can't hear it?
Yes, please. Sorry.
Sorry. Yeah. Hi. Thank you. How do you take this information and then mitigate or utilize it so that we can actually factually, I guess, fight Bayer or Monsanto? But equally, I've been working with a team which was in the semifinal for the X Prize Rainforest Biodiversity Conservation Prize, where we use eDNA as well as we use our microbes and fungi and the plant and the botany, as well as the species, animal species. So as I called it, my soup, my bucket of soup, what is it now we know the contaminants and the health risks and health facts around it? How do we take this research that you've done here under your spring for the plant protection and find solutions that are viable and also, I guess, mitigate against further contamination?
Maybe I can answer this question because what we showed now is a sprint first phase. So actually we are working with the stakeholder train I was showing in my first presentation with all the stakeholder groups on possibility of transition to sustainable farming systems. The first phase, what we did in the project, is to see what is the fact, what is the moment, what can we see, and what is the risk. Is the next phase now we are in, and then we will go for transition. And we do it now Europe-wide, and we hope that as well in Argentina this will be possible to work with different groups on possible transition possibilities, and as well the role of the different stakeholders in the process of transition.
The problem is the mitigation.
I can't hear you. Please talk into the microphone.
The challenge is always the cost, the laws around it, and then who is paying the farmer to do the transition. And so also as a microfinance investor and working with agriculture, how do we get people back to soil remediation or the model of the one-third, I'm a biodynamic gardener and agricultural trained farmer as well. So where is it in this transformation? Where are the subsidies? Who's going to help finance and work at cleaning and clearing up? Not just that, there's also the cross-contamination from the water of those that aren't cleaning up And so there's always this transformational issue. And then again, also replantation of the trees, which is very important. And the tree, the food forest, I would say. Thank you.
Maybe you would like to join us as a stakeholder, because we really will figure out this for different countries, but this would be very important that we will collect ideas how it is possible, how macroeconomics and the cost, as Peter showed, it's 150 billion euro per year for 10 European countries. So this is costs which are paid by society in this moment. So so how can we do this? We are working on this now with our colleagues who are experts in this transition and policies. And if you would like to join our stakeholder network, please talk with Kayode and Esperanza. You are warmly welcome. Exchange.
Thank you very much. I already have her information.
Perfect.
So thank you very much for this question. Just to complement, the first step is to do awareness. So what we are doing now, and we have also given workshops in each of the case study sites, so to give the information to the farmers. Like this, they can know what is happening in the soil. Most of the time, they don't know. So the first step is awareness. And after awareness and different levels of the stakeholders, a multi-platform stakeholders, then is when it takes place, this discussion also. So you are very welcome to the workshops with different, yes, thank you very much. Okay, so I think we go to the next presentation because
and the last one.
And the last one. So we have Nelson Abrantes from the University of Aveiro. So do you have already the? Yes. Okay, perfect. So the floor is yours, Nelson.
Okay, thank you, Esperanza. Hello, good morning. Hope you can see my presentation. Yes.
Yes.
Okay, so it's an honor for me to be part of this session, and following the previous presentation of my colleagues on the pesticide distribution and impacts, I will address the topic of impact of realistic pesticide mixtures on freshwater ecosystems. Okay. As you know, pesticides, often designed to protect our crops, they frequently reach the aquatic systems, namely by surface runoff, also leaching, spray drift, wind erosion, so different processes. And when pesticides reach the aquatic systems, they can disrupt the balance of the ecosystem. And they affect a wide spectrum of non-target organisms, from primary producers to top predators. Recent data from the European Environmental Agency reveals that 22% of the European surface water exceed the pesticide thresholds. So this underscored the urgent need to address this topic. And we know that current agricultural practices, they comprise multiple pesticide applications. Farmers apply formulations with several pesticides, and they often undergo multiple pesticides application along the year. This results in complex mixtures in our environment. A monitoring study conducted also by the European Environmental Agency, where they analyzed over 10,000 aquatic systems in European countries, revealed the presence of multi-pesticides in the analyzed sites, ranging from 10 to more than 100 pesticides in a single water body. So what we have in our aquatic systems is a cocktail of pesticides. Also, as presented by my colleagues, the sampling study that we conduct in Sprint projects, where we analyze 64 aquatic systems across 10 European countries and Argentina, revealed the presence of several pesticide residues in surface water. And we found in our aquatic systems 8 to 40 pesticides. Also important to point that we normally assess the risk of pesticides based on a single compound toxicity, and this is unrealistic. So considering the presence of complex mixtures in the real world, we are potentially underestimating the risks of pesticide mixtures for the aquatic ecosystems. And these situations raise particular concern based on the assumption that the exposure to multiple pesticides poses a higher risk as distinct compounds they can interact and leading to synergistic effects. So the questions that we raised in sprints were, are pesticide mixtures more toxic to aquatic organisms than single pesticides? And are the mixtures we found toxic to aquatic life? To address this question, the following assessment strategy was defined. So, based on the pesticides found in aquatic systems of each 11 case studies of sprint, we define a mixture composed by 5 pesticides that were selected based on the hazardous to aquatic species and the more frequent pesticides. These are the mixtures that we define for each case study. So you can see that most of them are composed mainly by herbicides and fungicides, but also some of them also include insecticides. So each of the mixture was tested at 5 concentrations, including a control where no pesticide was applied, and then we include the MEC, the measured environmental concentration, that is a realistic concentration. Also the PEC, the predicted environmental concentration, according to the EFSA, calculated by the Focus model, and we also defined the 3 and 5 PEC to establish a dose effect response. And important to note that all of these concentrations were below the toxicity values reported for each single compound. This based on DSC 50s from EFSA reports. So in total, we tested 5 aquatic species, including organisms from different trophic and functional levels. Okay, very briefly, the species that we tested was a macroalgae, Rhaphidocelis subcapitata, also an aquatic plant, Lemna minor. In both, we look at grow inhibition. We also test a pelagic invertebrate, the common Daphnia magna, and also a benthic invertebrate, Chironomus, where we look at immobilization after 48 hours. And finally, we also test zebrafish, where we look at survival after 96 hours. And moving now to the results, and let me start showing you the response of the microalgae. So in this graph, each bar corresponds to one of the 11 mixtures, and each color corresponds to the different concentrations. The number inside the colors means the percentage of effects that we observed in each concentration. So if we look at this graph, we clearly see that the microalgae was impacted in all mixtures. So a grow inhibition was observed in all them. The concentration PEC, 3 PEC and 5 PEC induced inhibitions higher than 30% in some mixtures. And also important to note that even the MEC, the realistic concentration, also induced a significant grow inhibition. And similarly to the microalgae, also the aquatic plant, if we look at the graph, we see that all the pesticide mixtures induced the grow inhibition. And in some mixtures, the inhibition was higher than 50% after exposure to the PEC, 3 PEC and 5 PEC. And once again, the realistic concentration, the MEC, we can see in the light green, also produced an inhibition of around 10% in some mixtures. Okay, for the invertebrate Daphnia magna, okay, the results show us that from the 11 mixtures, 8 of them induced lethal effects. And in some cases, the mortality was higher than 50% for some mixtures. And also important to note, sorry, that a total mortality, 100% was observed in some mixtures. And let me remember that all the concentrations tested were below the toxicity values for each compound. So we don't expect toxicity, but as you can see, in some mixtures, we have 100% of toxicity. Okay, and moving to the benthic invertebrate. Again, it was also severely impacted by the exposure to all mixtures. 3 and 5 pack in some cases induced again 100% of mortality, and again the realistic concentration induced in some mixtures an effect higher than 20%. Finally, for zebrafish, we observed, and compared with the other species, we observed a lower toxicity. The maximum effect was 25% of mortality that we observed in the highest concentration. Important also to say that the recent assays that we did with the sublethal endpoints, where we look at the cholinesterase activity, revealed that even at lower concentrations, we found effects. Okay, so let's move to the conclusions of this study. All tested species were impaired when exposed to the 11 distinct mixtures. In the case of producers and invertebrates, they were highly impacted. Zebrafish was the most tolerant in what concerns the acute toxicity that we access. For some mixtures, we observed a total effect with 100%, namely in the highest concentration. So we observed a severe effect. Really important to highlight that significant effects were observed at MEC, the realistic concentration, and this underlies the hazard posed by pesticide mixtures to aquatic ecosystems in the real environment. And we also observed that the toxicity of the mixture was dependent, of course, on the species that we tested and on the formulation of each mixture. For instance, in a mixture with more herbicides, it is expected that we have more effects on plants. So finally, let me leave you two core messages. Mixtures showed to be much more toxic than single compounds. So the combination of multiple pesticides had a more severe impact on aquatic organisms. And therefore, our findings highlight severe effects caused by pesticides mixtures on aquatic ecosystems, and of course, the urgent need to protect them, namely in agricultural areas. Thank you so much for your attention. If you have questions, I'm happy to answer.
Thank you very much, Nelson. Okay, so do we have questions here? Online? Do we have questions online? No? Okay, I have a question from Declan. He left, but he said to me, and it's mainly for Violette, I think, what is the message to take, the final message that the UN can take from
this comes now?
Exactly. So please.
To be now?
The final and very shortly is that our aquatic systems are not safe.
Very clear. And what I will now do, I made a small summary of all the recommendations, and that I was asked to do by for Declan. First of all, I would like to thank you all for the speakers, and as well, I got from Jose Luis Costa that you made an excellent presentation, Nelson. So that was done in chat. I thank you, the people who attended the meeting, a lot for your attendance. We will check whether we can give you this recording as a file. We will figure this out. So people who would be interested, just send a mail to Kayode or to Sprint, better to Kayode himself. So if you were interested in this recording, and we will figure out which part we can give to you. Okay, just to make a short summary about what we have heard today is we have several facts. The fact is, mixtures of pesticide residues are omnipresent in environmental and human matrices. We don't talk about single pesticides. We talk always about mixtures. Clear effects of selected pesticides, such as glyphosate, but as well others, on gut microbiome composition, gut cell stress, altering energy production, and gut barrier functioning is seen and clearly described. That means the pesticides we detect in the environment and in humans are as well mostly described to be hazardous, also the majority of them are described to be hazardous, and we could find clear effects on them, on human health. Glyphosate showed even at acceptable daily intake increases of anxiety-like behavior conducted by mice experiments in the University of Cork. What we saw is that benchmarks are missing for most matrices. That the atmospheric transport is not taken into consideration by EFSA, and in all transport models of EFSA, this very important issue is missing. And as Daniel said, for several pesticides, this environmental burden is much higher for exposure than the burden by uptake by food. Looking at costs, we could say that substantial remarked external costs of pesticides, pesticide use and human and environment health make current pest control practices unsustainable. And Peter estimated costs of 150 billion euro for the 10 European countries. So what do we need? First of all, urgently, we need to include a pesticide regulations by EFSA, EPA, or the other country-wide pesticide regulation organizations. Effects of mixtures on ecosystem and human health. As Nelson and Kayode could show, the mixtures have a much stronger effect than the single pesticide. Furthermore, as Maaike could show, sensitive indicators such as gut microbiome changes, the gut barriers, if reflecting the real effects of the real world, should be included into the regulation system. Furthermore, as Dirk and Freya showed, transport, aerial atmospheric transport is a very important issue. Therefore, transport models that are calibrated and validated on monitoring data in fields should be used for pesticide regulations. And include atmospheric transport models for pesticides transported by wind erosion over long distances. Define toxicological-based thresholds for pesticides, for single ones and their mixtures in air and in indoor dust. And define benchmarks of daily intake of residues mixtures related to the different exposure routes, and not only for food, because this does not reflect the reality. So what we come up with these results of the sprint, we can show that we are all exposed, we as well as the ecosystem, by mixtures which are characterized as hazardous. This causes health effects and costs. The question is, why do we still stick to an agriculture which is born after the World War II to produce as much food as possible and per hectare? What has led to this idea was the idea never having hunger again, which was a very valid idea, I think, after the World War, where a lot of people were starving and dying of hunger. But in our world now, we produce more than 4,000 calories per person per day. So it's not an idea that we do not produce enough food. It's a distribution problem. We waste 30% of our food. So we can go for a new modern agriculture. And this transition of the agriculture urgently, due to my opinion, has to be urgently done. And modern agriculture includes agroecological concepts, modern robotics, modern technologies. They are all there waiting to be implemented. So we can make the change towards sustainable food production on a global scale if we all try to do it and we all work on a transition, including consumers' awareness, taking industry with us to come up with ideas, governments, NGOs, to come up with ideas how to implement it, what is needed, and come up here with a global approach, strongly decreasing human and ecosystem health effects by agriculture, which is now conducted actually. Thanks a lot for your contribution, and I hope you enjoyed our session. I just wanted to say there were some questions from Argentina. I don't know whether these persons are still there or they already left, I think. They were asking to give some information on Spanish, but these people could contact me then later, and so maybe tomorrow or on Friday we could call and I give them some information in Spanish if it is needed. Yeah, we did not, there was a question as well why we did not ask specifically Virginia from Inter to present the results from Argentina. That is because we gave an overview of the results of all case study sites. So we did not zoom in into specific case study site in this session. And hopefully next year we can contribute to the UN Science Summit again, then having more ideas on transition and ways to transition. And thanks again for staying with us, and I hope you had an interesting morning or afternoon, depending where you are. Esperanza, do you want to say some final words?
Yes. Thank you very much, Violette. Thank you to all the presenters and also to the public, those here and also online. Thank you very much for being here. And together is the way that we can go further for the transition. Yes. Thank you very much to all. I will give a applause.
Okay. Yeah, so for those that need the recording, so just shoot me an email for those that need it, so we can probably get it available. So thank you once again. It's been a pleasure having everyone here. So we take one or two things home. Thank you.
Yeah.
Yeah,
thank you to all. And yeah, we keep in contact. So bye-bye.